Hello everyone, my name is He Luo from the Center for Quantitative Biology at Peking University, under the supervision of Professor Zhexian Zeng. Today, my presentation is titled Integrated Computational Analysis Identifies Therapeutic Targets with Dual Action in Cancer Cells and T cells. Over the past decades, cancer immunotherapy has developed at a remarkable pace. In 2014, the FDA approved the first PD-1 antibody for cancer treatment, and the discoverers of immune checkpoints, James Allison and Tasuku Honjo, were awarded the 2018 Nobel Prize in Physiology or Medicine. As shown on the right, the principle of immunotherapy is straightforward. Cancer cells often overexpress PD-L1, a ligand that binds to its receptor PD-1 on T cells. This interaction suppresses T cell activity, allowing the tumor to evade immune surveillance. Antibody blockade of PD-L1 tends to restore T cell function and enables durable anti-tumor responses. This strategy is known as immune checkpoint blockade therapy, or ICB for short. Because of its broad efficacy and long-lasting responses, ICB has become one of the most widely used immunotherapies in clinical practice. Beyond ICB, cellular therapies, most notably CAR T-cell therapy, constitute another major pillar of immunotherapy. Through genetic engineering, T cells are modified to express a chimeric antigen receptor, CAR for short, enabling them to specifically recognize and destroy cancer cells expressing the target antigen. A remarkable case is Emily Whitehead, the young girl shown on the right, who was diagnosed with acute lymphoblastic leukemia at age 6. She was the first child to undergo CAR T therapy for leukemia and has remained cancer-free for more than a decade, milestone in modern oncology. Today, CAR T therapy continues to bring hope to many patients with hematologic malignancies. However, when it comes to solid tumors, the effectiveness of immunotherapy remains limited, with objective response rates often below 10%. So, what drives these therapeutic resistances? In general, there are two main barriers to effective immunotherapy: external and internal. Externally, solid tumors build a complex, immunosuppressive microenvironment. It's rich in inhibitory signals and cells like regulatory T cells and tumor-associated macrophages. These factors hinder cytotoxic T cells from infiltrating the tumor and impair the immune response. Internally, even when T cells enter the tumor, they progressively become dysfunctional, marked by upregulation of inhibitory receptors like PD-1 and TM3, reduced production of effector molecules, and diminished proliferative capability. Therefore, identifying new therapeutic targets is key to overcoming these challenges. The scientific question I'm interested in is, can we identify dual-action targets, genes that, when inhibited, can both suppress tumors and boost T-cell immunity, thereby enabling a two-pronged therapeutic strategy? To discover such targets, we leverage CRISPR screening, a powerful tool for functional genomics and target discovery. CRISPR screening works by using a library of guide RNAs, where each sgRNA targets a specific gene. These sgRNAs are introduced into Cas9 expressing cells, creating a pool of cells with single gene knockouts. Exposing these edited cells to a selective pressure, such as co-culture with T cells, to identify which knockouts make the cancer cells more sensitive to T cell mediated killing. By sequencing the sgRNA barcodes and measuring their abundance, we can identify genes that enhance cancer cells' susceptibility to T cell killing, offering potential targets for improving cancer immunotherapy. Because CRISPR screening causally links genotype to phenotype, it's an efficient approach for mechanistic discovery and has enabled many high-impact studies. However, each screening experiment typically focuses on a single cell type and a single phenotype, making it challenging to directly pinpoint dual-action targets. To address this, I collected and standardized
558
immune-related CRISPR screening comparisons from 90 studies and developed a web-based platform called ICRAFT, which stands for Immune-Related CRISPR Screen Analyzer of Functional Targets. It's freely accessible online via this link. Using ICRAFT, researchers can easily explore how gene knockouts impact diverse phenotypes across different cell types. The platform has already been widely adopted by researchers worldwide. ICRAFT currently includes 397 screen comparisons from cancer cells across various cancer types, with 135 in vivo comparisons. Additionally, it includes 161 screen comparisons from 6 immune cell types. In addition, it integrates about 2 million single-cell RNA-seq profiles from tumor immune microenvironments and 943 bulk RNA-seq datasets from pre-immunotherapy samples. These datasets enable users to explore gene expression specificity across cell types and link candidate genes to clinical immunotherapy responses. With ICRAFT, researchers can now establish a systematic pipeline that integrates computational target prioritization with experimental validation. Effectively, integration of large-scale CRISPR datasets is crucial for analysis and deriving insights. Many regular analyses rely on taking intersections across multiple screens, which can handle only a limited number of datasets, typically no more than 6. In contrast, I ranked genes from good to poor based on their effect in each screen. I then applied the robust rank aggregation algorithm to integrate these rank lists, enabling scalable and robust integration across any number of screen datasets. Firstly, I integrated 34 CRISPR screens conducted on cancer cells under T-cell co-culture conditions in both human and mouse cell lines. The resulting cancer cell target list included positive controls, such as the anti-apoptosis protein CFLAR and TRF2, as well as novel candidates like PIGU and GPA1. Next, I analyzed 20 T-cell screens covering TCR activation, cell proliferation, and tumor infiltration phenotypes. This analysis produced a T-cell target list that included positive controls, such as the TCR signaling negative regulators CBLB and CD5, along with novel target candidates like CYC1 and UBE2F. By comparative analysis of the cancer cell and T-cell target lists, I identified 82 potential dual-action targets. Pathway enrichment and protein-protein interaction analyses revealed that these targets are enriched in lipid metabolism,
TNF and NF-κB pathways.
Among the 82 dual-action targets, SOCS1, TNF-IP3, and PTPN2 are the most prominent. Literature review confirmed that SOCS1 knockout can enhance both CD4 and CD8 cell function, while SOCS1 inactivation in cancer cells can reduce PD-L1 expression. Similarly, PTPN2 knockout could alleviate T-cell exhaustion, and loss of PTPN2 can sensitize tumor to immunotherapy in vivo. In contrast, evidence for TNF-IP3, also known as A20, acting as a dual-action regulator was limited, prompting us to validate it experimentally. TNF-IP3 encodes a ubiquitin editing enzyme that negatively regulates the TNF,
NF-κB,
and JAK-STAT pathways. We began experimental validation with cancer cells. TNF-IP3 knockout consistently increased their sensitivity to T-cell mediated killing across 3 mouse and 3 human cell lines. In vivo, TNF-IP3 deficient tumors exhibited slower growth, higher immune infiltration, and an enhanced response to anti-PD-1 therapy, suggesting synergy with immune checkpoint blockade. RNA-seq analysis revealed that TNF-IP3 loss upregulated ICAM1, CCL5, and other immune-related molecules, along with activation of TNF and
NF-κB
signaling pathways. Furthermore, TNF-IP3 deficient cancer cells were more sensitive to TNF-alpha-induced apoptosis, and this phenotype was rescued by deletion of the TNF-alpha receptor. These results indicate that TNF-IP3 loss enhances tumor killing by sensitizing cancer cells to TNF-alpha-mediated apoptosis. Taken together, our findings suggest that TNF-IP3 is a potential therapeutic target in cancer cells. Then, we further investigated the role of TNF-IP3 in T cells. In vitro co-culture assays, TNF-IP3 knockout in T cells enhanced their killing capacity, resulting in fewer surviving cancer cells. Additionally, TNF-IP3 knockout increased
interferon gamma
production in CD8 T cells. T-cell adoptive transfer assays demonstrated that loss of TNF-IP3 in T cells improved tumor control in vivo. RNA-seq analysis of TNF-IP3 null T cells after co-culture revealed upregulation of
NF-κB,
NF-κB1, NF-κB2 co-stimulatory molecules, OX40, HVM, as well as other genes involved in TCR signaling and effector function. Thus, TNF-IP3 functions as a negative regulator in both tumor and CD8 T cells. Having validated TNF-IP3 function in both compartments, we wondered what if we delete TNF-IP3 in both cancer cells and T cells, would the effects be enhanced? We performed cross-co-culture experiments by deleting TNF-IP3 in both cancer cells and OT1 T cells. The dual knockout group showed the fewest surviving cancer cells, significantly fewer than the two single knockout groups. In vivo, the dual knockout group exhibited the slowest tumor growth and highest immune infiltration, highlighting an enhanced anti-tumor immune response. Therefore, TNF-IP3 serves as a dual-action therapeutic target. I further explored its clinical relevance. Based on the differentially expressed genes from TNF-IP3 knockout in cancer cells, a TNF-IP3 deficient signature was constructed. Strikingly, a pan-cancer analysis revealed that the TNF-IP3 deficient signature was positively correlated with interferon response and inflammatory pathways, and negatively correlated with immune-suppressive pathways, such as M2 tumor-associated macrophages and exclusion, across most TCGA cancer cohorts. 4D convolution analysis consistently linked TNF-IP3 dysfunction to increased T-cell infiltration. Expression correlations showed positive associations between TNF-IP3 loss and ICAM1 and MHCI components, consistent with our experimental findings. Proteomic data further indicated a positive correlation between TNF-IP3 deficiency and LCK, along with other T-cell maturation markers. Moreover, in 3 ICB therapy cohorts, responders exhibited significantly higher levels of TNF-IP3, suggesting that patients with TNF-IP3 deficiency may respond better to immunotherapy. I curated a pan-cancer clinical cohort of over 30,000 patients and found that those harboring TNF-IP3 alternations had a significantly better prognosis. Given that TNF-IP3 is a well-known autoimmune susceptibility gene, we hypothesized that such alternations could enhance immune activity, thereby reducing cancer risk. To test this hypothesis, I curated around 20 autoimmune-related genes and performed Cox regression analysis, even after adjusting for confounding factors like age, sex, and cancer type. Patients with autoimmune-related alternations still showed a lower hazard ratio. This suggests that these alternations may activate immune system and reduce cancer risk. But this is still a direction that needs further investigation. Interestingly, in October 2023, Nature reported the first small molecule inhibitor of PTPN2, Its mechanism involves both making tumors more sensitive to immune attack and enhancing the function of T cells, NK cells, and dendritic cells. In preclinical models, its monotherapy efficacy is comparable to that of ICV, and it has already entered phase I clinical trials. This study provided independent validation of our dual-action target concept and its clinical potential. Finally, let me briefly introduce the ICRAFT platform. It's freely available for use through this link. It has 3 modules: Explore Gene, Explore Data, and Upload Data. In Explore Gene, you can query a gene of interest, choose the perturbed cell category, experimental condition, and screening data sets. After submission, ICRAFT then display the gene's knockout effects on immune phenotypes, expression differences between ICB responders and non-responders, and expression across cell types. Explore Data module enables integrated analysis. You can select desired data sets, and ICRAFT will automatically perform rank aggregation to generate candidate target lists. This function is designed to help researchers discover new functional genes. In Upload Data module, you can upload your own screenings and compare them with curated datasets in ICRAFT to identify consistent or context-dependent functional targets. To support the scientific community, all data in ICRAFT, including count matrices, analysis results, single-cell RNA-seq data, and immunotherapy bulk RNA-seq data, are openly available and freely downloadable. We hope these resources would help the community facilitate discovery and spark new ideas in functional genomics and tumor immunology. To summarize, we developed ICRAFT, a platform that enables systematic discovery of immune-related functional targets. Using it, we identified dual-action targets whose inactivation could make tumor more vulnerable while enhancing T-cell function. Through in vitro and in vivo experiments, we validated TNFAIP3 as one such target and elucidated its mechanism. Clinically, patients with TNFAIP3 dysfunction may show better responses to ICB therapy and improved prognosis. This work was published in Immunity in March 2025. Please feel free to scan the QR code to explore ICRAFT. We warmly welcome any feedback and hope that ICRAFT can help you identify meaningful targets. Our study was also highlighted by an immunity preview, which emphasized ICRAFT's ability to identify dual-action genes that both enhance T-cell anti-tumor immunity and sensitize tumors to immune attack. We also received encouraging comments during peer review. Looking ahead, we hope that ICRAFT will continue to provide tools and insights for tumor immunology and inspire new therapeutic strategies. I sincerely thank my supervisor, Professor
周贤珍、
and my co-advisor, Professor
登攀、
for their patient guidance and generous support. I also want to thank my collaborators, Dr. Rui Zhang and Rui Guo, who conducted most of the in vitro and in vivo experiments. Finally, I want to express my gratitude to all professors and colleagues who contributed to this work. Professor Zheng leads Zheng Lab with rigor and vision. Our team combines strong computational support with well-equipped experimental platforms in a collaborative environment. We warmly welcome talented students to apply and join Zheng Lab for cutting-edge and interdisciplinary research. Last but not least, let me share some reflections on this project. The idea originated from Professor Cheng's great insight. He proposed that let's collect and organize all publicly available CRISPR screens related to immunity, turn waste into wealth, rather than repeatedly performing similar experiments. We decided to systematically integrate existing functional evidence and let the data speak for itself. While collecting and standardizing CRISPR screening datasets, I learned a lot about experimental designs and gradually realized that computational approaches could uncover dual-action targets that are invisible in single experiment settings. In other words, computational methods can help turn data into knowledge and discovery. If such targets can enhance immunity while suppressing tumors, they may hold the key to overcoming the barriers in solid tumor immunotherapy. Of course, the journey was not easy. Curating metadata tables, unifying data standards, and mitigating batch effects. Every step was a challenge, but persistence paid off. We eventually solved the problems of standardization, robust integration, and visualization, trying to make ICRAFT a useful platform for the community. Collaboration matters. I'd like to especially thank Penjan for optimizing the ICRAFT website, improving its stability, speed, and user experience. The experimental validations were also very important. Without them, the dual-action targets would have remained conceptual. Special thanks to Dr. Wawala Everpertus, Rui Zhan, and Rui Guo, whose elegant cellular and animal experiments turned computational predictions into biological reality and elucidated the mechanisms of dual-action regulation. This project was truly a collaborative, cross-disciplinary effort. I've always believed that computation is a powerful lever. It can organize scattered evidence in big data into testable hypotheses, detect meaningful signals, and accelerate scientific discovery. Thank you all for listening. Any comments and suggestions are welcome. I hope that together we can make immunotherapy benefit more patients in the future.